Welcome, everyone. I'm excited to welcome you to today's presentation on mineral nutrition in plants. This is the cover slide, so I'll keep it brief. Today, we'll explore how minerals are essential for plant growth and development. We'll cover key nutrients, their functions, and how plants absorb and utilize them. Let's get started and dive into the fascinating world of plant nutrition. Let's take a closer look at the metabolism of nitrogen, which is a vital process for plant growth. Understanding nitrogen metabolism helps us see how plants convert atmospheric nitrogen into forms they can use, which is crucial for their development. Now, moving on to the toxicity of micronutrients, it's important to recognize that while these elements are essential in small amounts, excess can be harmful. Balancing micronutrient levels is key to healthy plant growth and avoiding toxicity symptoms. Next, we have the essential elements. These are the nutrients that plants absolutely need to complete their life cycle. Without them, growth is stunted and productivity drops significantly. Nodule formation is another fascinating topic. It involves the development of specialized structures on legume roots where nitrogen fixation occurs, thanks to symbiotic bacteria. This process is vital for enriching soil nitrogen content naturally. Soil as a reservoir of essential elements emphasizes the role of soil in supplying nutrients. Healthy soil contains a rich pool of minerals and nutrients that plants draw from throughout their growth cycle. Common deficiency symptoms are signs that plants show when they lack specific nutrients. Recognizing these symptoms helps us diagnose and correct nutrient deficiencies early. Criteria for essentiality refer to the standards used to determine whether an element is truly necessary for plant growth. These criteria help guide fertilizer development and nutrient management. Symbiotic biological nitrogen fixation is a process where certain bacteria convert atmospheric nitrogen into a form plants can use, working in tandem with plant roots. An incredible natural partnership. Translocation of solutes involves the movement of nutrients and other solutes within the plant, from roots to shoots and leaves, ensuring all parts receive what they need. Biological nitrogen fixation, as a broader concept, includes both symbiotic and free-living bacteria that convert nitrogen gas into ammonia, which plants can assimilate. Deficiency symptoms of essential elements are specific signs that indicate a particular nutrient is lacking, such as chlorosis or stunted growth, guiding us toward proper intervention. Mechanism of absorption of elements describes how plants take up nutrients from the soil, primarily through their roots, often involving specific transport systems. Essential mineral elements encompass both macro and micronutrients that are vital for various physiological functions within the plant. The role of macro and micronutrients highlights how each group contributes differently. Macronutrients are needed in larger amounts, like nitrogen and phosphorus, while micronutrients, such as zinc and iron, are required in trace amounts but are equally essential. Finally, methods to study the mineral requirements of plants include various experimental approaches. These help us understand exactly what plants need, how much, and under what conditions, so we can optimize their growth and productivity. These methods require purified water and mineral nutrient salts, which are essential for precise control of plant nutrition. Hydroponics, for example, has proven to be a successful technique for the commercial production of vegetables like tomatoes, seedless cucumbers, and lettuce. It's important to remember that the nutrient solution used in hydroponics must be adequately aerated, because oxygen availability is crucial for optimal plant growth. Interestingly, back in 1860, Julius von Sachs, a pioneering botanist, demonstrated something groundbreaking. He showed that plants could be grown to maturity in a defined nutrient solution without any soil at all. This was the first clear evidence that plants can thrive in a soil-less environment, and that technique is now known as hydroponics. Understanding the methods to study the mineral requirements of plants is fundamental because it helps us optimize growth conditions and improve crop yields. By controlling and analyzing the mineral content, we can better understand how plants absorb nutrients and how to meet their specific needs for healthy development. This connects directly to the overall theme of efficient and sustainable plant cultivation. Most of the minerals present in soil can enter plants through their roots. This process is crucial because it allows plants to absorb the nutrients they need to grow and develop properly. Interestingly, out of the 105 elements that have been discovered so far, more than 60 are found in different plants. Some plants have the ability to accumulate specific elements. For example, certain species can gather selenium, which is an essential trace element for humans and animals. Others might accumulate gold, which might sound surprising, but it's a real phenomenon in some plants. Additionally, plants growing near nuclear test sites can take up radioactive substances like strontium, specifically radioactive strontium. These examples highlight the diverse ways plants interact with and absorb elements from their environment, which ties into the broader theme of how plants play a vital role in the cycling of elements in ecosystems. Let's start by looking at the criteria for an element to be considered essential for plants. First, it must be directly involved in the plant's metabolism. This means the element plays a role in key biochemical processes. Next, the requirement for this element must be specific. In other words, no other element can replace it. If the plant is deficient in this element, simply adding more of a different one won't solve the problem. Third, the element has to be absolutely necessary for supporting normal growth and reproduction. Without it, the plant can't complete its life cycle or set seeds, which shows how critical it is. Finally, these points together form the key criteria for determining whether an element is essential. They help us understand why certain elements are vital for plant health and development. This concept is fundamental as we explore how plants acquire and utilize nutrients. Now, let's take a closer look at macronutrients and micronutrients. These are the essential elements our bodies need to function properly. Macronutrients include carbohydrates, proteins, and fats, the nutrients we need in larger amounts to provide energy and support bodily processes. Micronutrients, on the other hand, are vitamins and minerals, required in smaller quantities, but just as vital for health. Moving on to essential elements, these are specific nutrients that our bodies cannot produce on their own, so we must obtain them through our diet. They are crucial for maintaining overall health, supporting immune function, and ensuring proper growth and development. Understanding these categories helps us appreciate how balanced nutrition plays a key role in our well-being. By focusing on both macronutrients and micronutrients, along with essential elements, we can make informed choices to optimize our health and prevent deficiencies. Let's take a closer look at some of the essential macronutrients that plants need in large amounts. On this slide, you'll see a list of key elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. Now, what's interesting is that carbon, hydrogen, and oxygen are primarily obtained from CO2 and H2O, so these are the building blocks for most organic molecules in plants. The other nutrients, like nitrogen, phosphorus, and the others, are absorbed from the soil as mineral nutrients. These elements are present in plant tissues in large quantities, more than 10 millimoles per kilogram of dry matter. That's why we call them macronutrients. They're fundamental for plant growth and development. Understanding how plants access and utilize these nutrients is key to understanding plant health and productivity overall. Now let's take a closer look at some of these essential elements. We have boron, copper, nickel, zinc, manganese, chlorine, molybdenum, and iron. These elements are needed in very small amounts, less than 10 millimoles per kilogram of dry matter. Despite their small quantities, they play critical roles in plant health and development. These are often referred to as trace elements or micronutrients. Even though we only need tiny amounts, their presence is vital for processes like enzyme function, nutrient uptake, and overall growth. Recognizing these micronutrients helps us understand that a balanced supply of all nutrients, big and small, is key to healthy plant productivity and ultimately successful farming. Now, moving on to slide 9, let's look at how essential elements are categorized. These elements are grouped into 4 broad categories based on their diverse functions within plants. This classification helps us understand their specific roles in plant growth and development. It's fascinating to see how each group contributes uniquely to the overall health of the plant. For instance, some elements are crucial for structural components, while others are involved in metabolic processes. Recognizing these categories allows us to better grasp the complex nutritional needs of plants and how they utilize different elements for optimal functioning. Now, let's talk about some essential elements that can influence the osmotic potential of a cell. For example, potassium plays a crucial role in the opening and closing of stomata, which are vital for gas exchange and water regulation in plants. This shows how specific elements can directly impact cellular functions. Moving on, maintaining osmotic potential also involves elements that activate or inhibit enzymes. Take magnesium, for instance, which acts as an activator for ribulose bisphosphate carboxylase, an enzyme necessary for photosynthesis. Additionally, essential elements are key components of energy-related chemical compounds within plants. Magnesium is part of chlorophyll, enabling photosynthesis, while phosphorus is fundamental in ATP, the energy currency of the cell. Beyond that, these elements are also integral to the structure of biomolecules, forming the structural elements of cells themselves. Carbon, hydrogen, oxygen, and nitrogen make up the backbone of many vital molecules, providing the framework for cellular architecture. Understanding these categories helps us see how essential elements are interconnected with the overall health and functionality of plant cells, supporting processes from energy production to structural integrity. Now, let's delve into the role of these essential minerals for plants. Starting with chlorine, it is crucial for the water-splitting reaction in photosynthesis, which ultimately leads to oxygen production. Chlorine, along with sodium and potassium, helps maintain the solute concentration and the balance of anions and cations inside the cells, which is vital for cellular function. Plants absorb chlorine mainly as chloride ions, Cl-, from the soil, and it's needed everywhere in the plant body. Moving on to molybdenum, it plays a key role in nitrogen metabolism. It is a component of enzymes like nitrogenase and nitrate reductase, which are essential for nitrogen fixation. Plants absorb molybdenum as molybdate ions, Mo22+, and it is required throughout the plant, especially in leaves and seeds. Next, boron is involved in cell elongation, differentiation, and the translocation of carbohydrates through the phloem. It also facilitates cell membrane functioning and pollen germination. Boron is absorbed as borate ions, Bo33 or B4072, and is necessary mainly in the leaves and seeds. Copper is another important element. It is reversible oxidized from Cu+ to Cu2 during electron transport, making it essential for both photosynthesis and respiration. Copper also works with certain enzymes involved in redox reactions, supporting overall metabolism. Plants absorb copper as cupric ions, Cu2, and copper is needed everywhere in the plant. Zinc is required for enzyme activation, especially those involved in photosynthesis, respiration, and nitrogen metabolism. It also plays a role in the synthesis of auxin, a plant hormone regulating growth. Zinc is absorbed as Zn2 ions from the soil and is needed throughout the plant. Manganese is vital for water splitting during photosynthesis, releasing oxygen. It activates enzymes like catalase and is an important component of proteins like ferredoxin and cytochromes involved in electron transfer. Plants absorb manganese as Mn2 and need it primarily in leaves and seeds. Iron is involved in forming chlorophyll and activating enzymes like catalase. It also participates in electron transfer processes within the plant. Iron is absorbed mainly as ferric ions, Fe3, and is required everywhere in the plant. Sulfur is a part of amino acids like cysteine and methionine, essential for protein synthesis. It's also involved in DNA and RNA synthesis and activates enzymes of respiration and photosynthesis. Plants absorb sulfur as sulfate ions, SO42, and it's particularly needed in young leaves and meristematic regions. Magnesium is crucial for chlorophyll formation and plays a significant role in regulating metabolic activities. It activates specific enzymes and is involved in maintaining cell membrane integrity. Magnesium is absorbed as Mg2 and is needed in all parts of the plant. Calcium helps maintain cell turgidity, activates enzymes, and is involved in opening and closing stomata. It is essential for protein synthesis and maintaining ion balance within cells. Plants absorb calcium as Ca2, especially in meristematic tissues, buds, and root tips. Potassium is a key component of cell membranes, proteins, nucleic acids, and nucleotides. It's necessary for phosphorylation reactions and helps regulate osmotic pressure. Potassium is absorbed as K+ ions and is vital for overall plant health. Phosphorus is involved in forming nucleic acids, ATP, and other energy-related molecules. It supports cellular growth, division, and metabolic activities. Plants mainly absorb phosphorus as phosphate ions, H2PO4- or HPO42, and it's required in all parts of the plant. Finally, nitrogen is perhaps the most abundant and essential nutrient. It is a major part of amino acids, proteins, nucleic acids, vitamins, and hormones. Nitrogen supports all major metabolic processes like photosynthesis and respiration. It is mainly absorbed as nitrate ions, NO3-, but can also be taken up as nitrite or ammonium. It's required universally, especially in meristematic tissues and actively growing cells. Understanding these macro and micronutrients helps us appreciate how plants maintain their growth, development, and overall health, which is at the core of our overall theme of plant nutrition. The deficiency symptoms in plants can vary depending on the specific element that is lacking. These symptoms are usually visible as morphological changes, which we often refer to as deficiency symptoms. It's important to note that these symptoms will typically disappear once the deficient mineral nutrient is supplied back to the plant. However, if the deficiency continues over time, it can become severe enough to cause the plant's death. Now, whenever a particular element is present below a certain critical level, we say that the element is deficient. This critical level is key because it marks the point where the plant's growth starts to be affected. When the supply of an essential element becomes limited, the plant's growth slows down or gets retarded. This specific level of the element, below which growth is affected, is called the critical concentration. Understanding these deficiency symptoms is crucial because they help us diagnose nutrient problems early and take corrective action. It connects directly to our broader goal of ensuring healthy plant growth and maximizing productivity. Let's talk about how deficiency symptoms tend to show up in plants. Typically, these symptoms appear first in the young tissues, especially when the elements involved are relatively immobile. This means they don't get transported out of the mature organs easily. For example, elements like sulfur and calcium are integral parts of the cell's structural components, so they are not readily released or moved around within the plant. On the other hand, for elements that are actively mobilized, meaning the plant moves them around to support growth, deficiency symptoms usually show up first in the older tissues. Think of nitrogen, potassium, and magnesium. They are mobilized from older leaves to younger ones when needed. Consequently, when there's a deficiency, you'll notice the symptoms first in the senescent or aging leaves. This happens because the plant breaks down biomolecules containing these elements in the older leaves, making these nutrients available for transport to the younger, developing tissues. So, understanding whether a deficiency appears first in young or old tissues really depends on how mobile that element is within the plant, which ties into the overall theme of nutrient mobility and deficiency management. Let's take a closer look at some common deficiency symptoms in plants. First, when plants are lacking nitrogen, sulfur, or molybdenum, they often show delayed flowering. This means the plant takes longer to produce flowers, which can affect overall yield and reproduction. Next, deficiency of nitrogen, potassium, sulfur, or molybdenum can lead to the inhibition of cell division. This process is crucial for growth, so when it's hindered, the plant's development is stunted. Moving on, tissue death, especially in the leaves, is another symptom. This is called necrosis, and it usually occurs when the plant is short of calcium, magnesium, copper, or potassium. You'll notice dead patches or browning in the leaf tissue. Then there's chlorosis, which is the loss of chlorophyll, causing leaves to turn yellow. Nutrients like nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc, and molybdenum play a key role here. When they are deficient, the plant can't produce enough chlorophyll, leading to this yellowing. These symptoms are important clues for diagnosing nutrient deficiencies and ensuring proper plant health. Recognizing these signs early can help us take corrective actions to keep plants thriving. Therefore, excess manganese can actually cause deficiencies of other essential nutrients like iron, magnesium, and calcium. This means that what we might initially think are symptoms of manganese toxicity could actually be signs of deficiencies in these other minerals. For example, one of the main symptoms of manganese toxicity is the appearance of brown spots on the plant, which are surrounded by chlorotic veins. Manganese competes with iron and magnesium for uptake by the plant, and it also competes with magnesium for binding with enzymes. Additionally, manganese can inhibit calcium translocation in the shoot apex, affecting overall growth. Many times, when there's an excess of one element, it can inhibit the uptake of another, leading to complex deficiency symptoms. It's also important to note that if the concentration of a mineral ion in plant tissues reduces the dry weight of the tissue by about 10%, we consider that to be toxic. Remember, micronutrients are required in very small amounts, but even a moderate decrease can cause deficiency symptoms, while a moderate increase can lead to toxicity. So, maintaining the right balance of these micronutrients is crucial for healthy plant growth and development. In the second phase of uptake, the ions are taken in slowly into the inner space of the cells, which we refer to as the symplast. This phase is more controlled and involves the movement of ions through the cytoplasm and across the cell membranes. Now, if we look back at the first phase, it's characterized by a rapid uptake of ions into the free space, or outer space of the cells, known as the apoplast. This initial phase is passive, meaning it doesn't require energy, and ions move quickly into the apoplast right after they come into contact with the root surface. Much of our understanding about how plants absorb elements comes from studies on isolated cells, tissues, or organs. These experiments have helped us see that absorption happens in two main phases, which are crucial for understanding how nutrients get from the soil into the plant. So, to connect this to the broader picture, understanding these two phases helps us grasp how plants efficiently take up essential nutrients while also regulating what enters the inner parts of the plant. This dual-phase process is fundamental to how plants maintain nutrient balance and overall health. Let's talk about how ions move, which we usually refer to as flux. When ions move into the cell, we call that influx, and when they leave the cell, it's efflux. Now, it's important to understand that getting ions in or out of the symplast, the network of living cells, requires energy. This is what we call an active process, meaning it consumes metabolic energy to happen. On the other hand, ions can also move passively, especially into the apoplast, which is the space outside the cell membranes. This passive movement often occurs through specialized structures called ion channels. These channels are transmembrane proteins that act like selective pores, allowing certain ions to pass through while blocking others. Understanding these different modes of ion movement is crucial because it underpins how plants absorb nutrients and regulate their internal environment. Whether the process is active or passive, each plays a vital role in the overall process of element absorption, helping plants maintain their health and growth. This has been proved by the use of radioisotopes of mineral elements that they are transported through the xylem. When we analyze the xylem sap, it shows the presence of mineral salts in it. This sap is pulled up through the plant by a process known as transpirational pull. Essentially, as water evaporates from the leaves, it creates a negative pressure that pulls this mineral-rich sap upward. Mineral salts are transported through the xylem along with the ascending stream of water, ensuring that nutrients reach all parts of the plant. It has been conclusively demonstrated that the xylem serves as the main pathway for the translocation of minerals within the plant. This process is crucial for distributing essential nutrients from the roots to the leaves and other parts, supporting overall plant health and growth. Now, let's explore the crucial role of soil as a reservoir of essential elements. First, soil acts as a matrix that stabilizes the plant, providing a physical foundation for growth. It also harbors nitrogen-fixing bacteria, which are vital because they convert atmospheric nitrogen into a form that plants can use, effectively increasing the soil's nitrogen content. This process is key to maintaining healthy plant growth, especially since nitrogen is a major nutrient. Since deficiencies of essential minerals can seriously impact crop yield, farmers often need to supply these nutrients through fertilizers. But soil isn't just about nutrients. It also holds water and supplies air to the roots, creating the perfect environment for roots to absorb what they need. Additionally, soil is teeming with microorganisms that play various roles, from breaking down organic matter to facilitating nutrient cycling. Beyond mineral supply, soil performs many other important functions. For example, physical processes like weathering and the breakdown of rocks enrich the soil with dissolved ions and inorganic salts. These minerals, derived from rock minerals, are essential for plant nutrition and are thus called mineral nutrients. In essence, soil is a rich reservoir of all the mineral elements necessary for the proper growth and development of plants. It's composed of a wide variety of substances that work together to support healthy plant life. Recognizing soil's role as a reservoir helps us appreciate its importance in agriculture and ecosystem health. Now, apart from carbon, hydrogen, and oxygen, nitrogen is actually the most abundant element in living organisms. It plays a crucial role because it's a key component of amino acids, which are the building blocks of proteins. Nitrogen is also found in hormones, chlorophyll molecules, and many vitamins that are essential for life. In nature, plants and microbes are constantly competing for nitrogen, especially since the amount available in the soil is limited. This makes nitrogen a limiting nutrient, meaning it often controls the rate of growth and productivity in both natural ecosystems and agriculture. Most nitrogen exists as a very strong triple covalent bond between two nitrogen atoms, which makes it quite stable and not readily available for biological use. That's why nitrogen has to go through various transformations to become accessible to living organisms. In the nitrogen cycle, we see several key processes. Plants and microbes can take up nitrogen in different forms, such as ammonium or nitrate. Decaying biomass releases nitrogen back into the soil through a process called aminification. Biological nitrogen fixation is another vital process, either carried out by bacteria that convert atmospheric N2 into ammonia, or through industrial and electrical methods to produce usable forms of nitrogen. Then, we have processes like denitrification, where microbes convert nitrates back into atmospheric N2, completing the cycle. Understanding this cycle highlights how nitrogen moves through the environment, supporting life, while also emphasizing the delicate balance required for healthy ecosystems. Let's talk about some of the sources of nitrogen oxides in the atmosphere. These include industrial combustions, forest fires, automobile exhausts, and power-generating stations. All of these activities release nitrogen oxides into the air, which can contribute to air pollution and environmental issues. Now, shifting focus to natural processes, there's something called atmospheric nitrogen fixation. This is where lightning and ultraviolet radiation provide enough energy to convert nitrogen molecules in the air into nitrogen oxides, such as NO, NO2, and N2O. These natural processes play a significant role in the nitrogen cycle. Speaking of nitrogen fixation, it's important to understand that this is the process where atmospheric nitrogen, or N2, is converted into ammonia. This transformation is crucial because it makes nitrogen available to plants and other organisms. Finally, the first step in this process is called nitrogen fixation itself. It involves breaking the strong bond in nitrogen molecules and turning N2 into a form that can be used biologically. This step is essential for maintaining the balance of nitrogen in our ecosystems and supporting life. Now, the process of aminification is carried out by certain microorganisms, such as Bacillus ramosis and Bacillus vulgaris. These tiny organisms play a crucial role in breaking down organic nitrogen found in dead plants and animals. Essentially, they decompose the complex organic nitrogen compounds into simpler forms, primarily ammonia. This transformation is what we call aminification. It's a vital step in the nitrogen cycle because it converts organic nitrogen into inorganic ammonia, which can then be used in subsequent processes like nitrification. So, through this process, microorganisms help recycle nitrogen, ensuring it remains available in the ecosystem for new plant growth and maintaining the balance of the nitrogen cycle as part of the larger picture of nutrient cycling. Let's talk about nitrification, which is a crucial part of the nitrogen cycle. First, these nitrifying bacteria are chemoautotrophs, meaning they obtain energy by oxidizing inorganic substances, not by consuming organic matter. In the first step of nitrification, ammonia is converted into nitrite. This process is carried out by bacteria such as Nitrosomonas or Nitrococcus. Essentially, ammonia reacts with oxygen to produce nitrite, along with some protons and water. Some of the ammonia actually escapes into the atmosphere through volatilization, but most of it is transformed into nitrate by soil microbes. This whole process of converting ammonia into nitrate is called nitrification, and it happens in two main steps. Now, in the second step, the nitrite is further oxidized into nitrate with the help of bacteria like Nitrobacter. This reaction involves nitrite combining with oxygen to form nitrate. The nitrate produced is then absorbed by plants and transported to their leaves. Inside the leaves, the nitrate is reduced to form ammonia, which eventually becomes part of amino acids, the building blocks of proteins. So essentially, nitrification links ammonia in the soil to plant nutrition, completing an important cycle that supports plant growth and ecosystem health. Now, let's talk about denitrification. This process is carried out by specific bacteria, mainly Pseudomonas and Thiobacillus. These bacteria play a crucial role in the nitrogen cycle by converting nitrate, which is present in the soil, into nitrogen gas. This step is important because it helps remove excess nitrates from the soil, preventing issues like leaching and water pollution. Essentially, denitrification reduces nitrates back to atmospheric nitrogen, completing the cycle. By understanding this process, we see how bacteria help maintain a balance in soil nutrients and support healthy ecosystems. This step ties into the overall cycle of nitrogen transformation and highlights the vital role of microorganisms in environmental processes. Let's talk about biological nitrogen fixation now. As you can see, some microbes are capable of fixing nitrogen either freely or through symbiotic relationships. For example, cyanobacteria like Anabina and Nostoc are free-living nitrogen fixers, and they play a vital role in enriching the soil with nitrogen. Then, there are other microbes like Bacillus and Rhodospirillum, which are anaerobic microbes. They thrive in environments without oxygen and also contribute to nitrogen fixation. On the other hand, some microbes are aerobic, meaning they require oxygen, and examples include Azotobacter and Bajornichia. These microbes are also capable of fixing nitrogen on their own. It's important to understand that very few living organisms can utilize atmospheric nitrogen directly, because N2 in the air is quite inert. Only certain prokaryotes, like these microbes, can convert nitrogen in the air into a form that plants can absorb as ammonia. This process is called biological nitrogen fixation, and it's crucial because it makes nitrogen available to the entire ecosystem. The key enzyme involved here is nitrogenase, which is only found in prokaryotes. This enzyme reduces nitrogen gas into ammonia, enabling these microbes to fix nitrogen efficiently. So, in essence, these nitrogen-fixing microbes are vital players in maintaining the nitrogen cycle and supporting life as we know it. Let's take a closer look at some of the key plants involved in nitrogen fixation, starting with broad beans, lentils, and sweet clover. These plants are excellent examples of how certain species can form beneficial relationships with nitrogen-fixing microbes. Now, an interesting fact is that the microbe Frankia also produces nitrogen-fixing nodules, but unlike rhizobium, it can associate with non-leguminous plants, such as ulmus or alder trees. Both Frankia and rhizobium are naturally found in the soil, and as symbionts, they have the ability to fix atmospheric nitrogen, converting it into forms plants can use. Most commonly, these microbes form nodules on the roots of their host plants. These nodules are essentially small outgrowths on the roots that house the bacteria, creating a specialized environment for nitrogen fixation to occur efficiently. You'll notice many of these plants, clover, beans, garden peas, sweet peas, and alfalfa, are part of this symbiotic relationship. The most prominent and well-studied example of this is the legume-bacteria relationship. Here, species of rod-shaped rhizobium bacteria form close associations with the roots of legumes, facilitating nitrogen fixation. This process, known as symbiotic biological nitrogen fixation, is crucial because it naturally enriches the soil with nitrogen, reducing the need for chemical fertilizers and supporting sustainable agriculture. Overall, these partnerships are a fascinating and vital part of how ecosystems maintain their balance and productivity. Let's now look at the key role glutamic acid plays in amino acid metabolism. Glutamic acid is the primary amino acid involved in the transfer of the amino group, or NH2, which is crucial for forming other amino acids through a process called transamination. This reaction is catalyzed by an enzyme known as transaminase. Essentially, transamination involves transferring an amino group from one amino acid to the keto group of a keto acid, facilitating amino acid synthesis. Moving on, the process of transamination is fundamental because it allows the body to efficiently reuse amino groups, maintaining amino acid pools. When we see the reaction involving alpha-glutaric acid, ammonium ions, and NADPH, it results in the formation of glutamate, water, and NADP. This is a key step in amino acid synthesis. Now, let's connect this to nitrogen fixation in bacteria. When bacteria are released from the thread into plant cells, this leads to the differentiation of specialized nitrogen-fixing cells. During this process, ammonia reacts with alpha-ketoglutaric acid to form glutamic acid, which is vital for nitrogen assimilation. Focusing on ammonia at physiological pH, it exists as ammonium ion, NH4+. While plants can take up nitrate and ammonium, ammonium is actually toxic if it accumulates, so plants have mechanisms to manage this. Most microbes that fix nitrogen are aerobes, but switch to anaerobic conditions during nitrogen fixation to protect the nitrogenase enzyme, which is highly sensitive to oxygen. This enzyme requires a significant energy input, about 8 ATP molecules per ammonia molecule produced, energy that comes from the respiration of the host plant cells. The enzyme nitrogenase, a molybdenum iron protein, catalyzes the conversion of atmospheric nitrogen into ammonia, which is the first stable product of nitrogen fixation. This process occurs in specialized structures called nodules, which have adaptations like leg hemoglobin, an oxygen scavenger, to protect the enzyme from oxygen damage. The overall nitrogen fixation reaction involves nitrogen, electrons, protons, and ATP, producing ammonia, hydrogen gas, and regenerating ADP and inorganic phosphate. Inside the nodules, bacteria transform into rod-shaped bacteroids, which induce the division of inner cortical and pericycle cells, leading to nodule formation. This nodule then establishes a direct vascular connection with the plant, facilitating nutrient exchange. The formation of nodules begins with bacteria multiplying and attaching to root hairs, creating infection threads that carry bacteria into the root cortex, where they contribute to nodule development. So, in summary, nodule formation is a complex but highly coordinated process involving multiple interactions between rhizobium bacteria and the plant roots. ultimately leading to a mutually beneficial relationship that supports nitrogen fixation and plant nutrition.